Hello listeners, I'm Abigail Barton, and I'll be doing a presentation on Lynn Yibin and his research misconduct. Background on Lynn Yibin: he was a postdoctoral researcher, assistant professor at University of Texas Medical School. He was awarded an EAI Fellow in 2019. He had 6 publications and 8 manuscripts, which were all retracted due to his misconduct. I did a little more research on this guy than I needed to, but it was so interesting because I could not find him anywhere. He's pretty much erased from the internet and LinkedIn, probably due to his misconduct. I'm 98% sure this guy on the right is Lynn Yibin. However, I am not totally correct. Lynn Yibin did have a lot of retracted articles. However, we're only going to focus on one for this presentation. The one we're going to focus on for this presentation, seen on the right, that was withdrawn from bioarchives, was titled Effective Methods for Genomic DNA Mutagenesis in E. coli. The motive for this research is that current methods cannot induce multiple mutations as well as long DNA fragments, deletions, insertions, or replacement mutations. The scientific community kind of tries to accommodate for this. However, it requires specially designed primers, and you can only do certain types of site-specific mutagenesis. And most article found out that these result in higher false positives caused by primer dimmers, which the article states. So his proposal was to use a modified extension method of PCR method, polymerase chain reaction, to invoke a simpler yet effective method. Due to his misconduct, this was not true. However, it suggests that it can do multiple desired mutations on genes, and these included deletions, insertions, and substitution mutations. The article uses E. coli, kind of like a test subject, you can think. However, I researched what this was kind of used for in other articles. Other articles say that it's commonly used for to maintain and amplify small plasmid DNA, which is what they used it for to test his method. The methodology of this research is extensive. However, bear with me. The first step in this research was vector purification, which is commonly used to act as a carrier for desired DNA sequence in a host cell. This study did purification through gel erythropoiesis, restricted enzymes, DPN1, and a purification kit to eliminate any contaminants. The second step in this methodology is single-site mutations. The study used an ERRC8 gene from the E. coli, which was used to amplify by a cDNA being inserted into a PET30A, which is a bacterial vector for expression between restricted recognition sites on the E. coli and the recipient's plasmid. This study used 4 primers, a forward primer, a reverse primer, primer for positive strands with mutated bases, and a complementary primer. These were used to produce DNA fragments that could come together to form a full-length gene with expected mutations. The third step in this methodology was to induce the mutation at a specific location. A 2-step PCR method was performed at final volume. The final volume was denatured with PCR twice, then primered annealing at 60 to 50 degrees Celsius, followed by 10 cycles of annealing temperatures for DNA synthesis. Once the desired mutations were obtained, they were ligmentated into a backbone vector and incubated. Heat shocked, then incubated again overnight. 8 samples were then collected using DNA sequencing and tested using DNA sequencing. However, the study claims that all 8 samples showed mutant genus containing expected mutations, which is kind of suspicious that all 8 samples showed mutant genus. The step in this methodology was multi-site mutant genus with the ERCC8 proteins. 13 cysteines were replaced by serine to get a cysteine-free protein by primer pairs. 7 PCR reactions were performed for amplification of DNA fragments for desired multi-site mutations. Once these were obtained, they were subcloned into a backbone vector, then transformed into a DH5 alpha component cell. A colony PCR method was used to determine the existence or absence of inserted DNA in plasmid. Step in this methodology was replacement, insertion, and deletion of mutagenous. The procedures for these are similar to the ones previously described, using PCR reaction and primer pairs. However, these use a 200 BP DNA fragment to replace, insert, and delete from that E. coli gene, which was the ERCC8. Though they differ in PCR reactions, so I'm going to go over that. So replacement used 3 parallel PCR reactions. Insertion used the same 3 parallel PCR reaction, and deletion differed with it using only 2 parallel PCR reactions. In 6 papers and 8 manuscripts, Lin Yibin admittedly committed misconduct. He fabricated data from previous research. He falsified data, so made it so that it would show a positive result. He also committed plagiarism from previous articles and submitted fake authors to disguise himself and hide himself, probably from the misconduct he performed. Some of the consequences for Lin Yibin's actions were implemented on the basis of 2 CFR Part 180 and Part 376. It prohibits him from working on any project that includes government funding for 10 years. In this 10 years, he can also not serve on any advisory board or peer review. And of course, all publications were retracted. Authors' careers can be ruined with misconduct, as we've seen with Lin Yibin's suspension, loss of a job, and funding loss, and a stigma attached to his name, probably for life. Rejoining the scientific community after misconduct can be proven difficult, as colleagues will have a lack of trust and not be willing to work with that researcher. The university's reputation could also be ruined, as they are responsible for overseeing research through their IRBs for ethical misconduct. Therefore, intelligent scientists may not want to work at the institution, resulting in lower quality research in the university. And depending on the misconduct, the university could be facing a lawsuit. The scientist community is always affected when it comes to misconduct, as it produces a lack of trust in scientific data to the public, as we see in COVID-19 with the public not trusting scientists' words. Co-authors are also affected, as the study states in 2016, that all co-authors, even the lowest, experience the impact of misconduct. They experience lower publication flow in their own work, and also drops in citation rates due to stigmatization by association. And public effect, in the case of Lin Yibin's research, it could have affected agricultural field, as mutagenesis is used to produce plant mutations such as rice in Vietnam, China, and United States. This method, if not stopped, could have caused financial harm and/or loss in production of food. Regardless, research in general guides public action, knowledge, and trust, resulting in public consequences if false data is produced. Thank you for listening. I know I went a little bit over, but these are my references, as you can see, and the images references as well on a separate page. Thank you for listening.